Imagine this. You have a bucket of money, but no time, and you want to build brand new knowledge. Who would you want to lead such a creative expedition? An expert who has made their career in a well-studied silo of knowledge? Or someone who has ventured into new areas of research in between silos and worked across many domains?
The research that I've wanted to do since my undergrad encompasses many different silos. But there have always been these powerful voices in research saying you can only be successful if you establish your expertise, and maybe once you get tenure, you can cross disciplines and do multidisciplinary research. But the problem is that curiosity doesn't segregate into disciplines, and neither does the natural world.
The current academic system is built on incentives that often value work in pre-established disciplines more than they value exploration. Within silo research is more highly cited and better funded, but the most new knowledge is often uncovered in the space between fields. Let's talk to someone working in between the silos.
If we think about the most pressing challenges of our times, things like climate change or epidemics, these problems encompass many different silos. So, throughout my career, I chose to get training in many different disciplines: molecular biology, neuroscience, biophysics, evolutionary biology, so that I could perform and also lead multidisciplinary research. My lab is working on social exchanges, the materials they transmit, the impacts that these materials have on receivers, and how these systems evolve. Now, this work requires knowledge from multiple different domains: biochemistry, physiology, phylogenetics, social evolution, macroevolution, ecology, sensory neuroscience, development, collective behavior, network science. Social fluids tend to only be exchanged in critically important behaviors, things that involve both cooperation and conflict, like mating or parental care. The social fluid that we're studying is passed mouth to mouth in ant colonies through a behavior called trophallaxis. Now, we found that ants aren't just passing food in trophallactic fluid, but also growth hormones, nestmate recognition signals, and even bits and pieces of their own physiology that in normal animals simply remain inside. We're particularly excited about the proteins involved in developmental control, because these could act like ballots to determine the developmental fate of the larvae. So this means the colony could be using this form of social fluid exchange as a kind of method of voting through feeding, collectively deciding the fate of the larvae that they rear. It also means that the further an individual is from the larvae in the social network, the less strong of a vote she has. And this would mean that nurses, the youngest individuals in the colony, have the strongest vote.
We are building a system to track adults and larvae in a colony and the trophallactic exchanges that occur amongst them. Why? Unlike solitary animals, in social insects, developmental rates are highly variable because they are controlled socially through feeding. Now, is there a preferential treatment given to certain larvae? What controls whether or not a larva gets fed? Do larvae beg? Could some workers actually be the production centers for developmental factors that impact larval growth?
Can ants perceive the quality of the trophallactic fluid they receive? In other words, can they read their nestmates' ballots before passing them on? By giving ants the choice between real trophallactic fluid and artificial trophallactic fluid that we create, we can see if ants are able to perceive certain compounds within the trophallactic fluid. Do ants with different roles in the colony have different abilities to sense trophallactic fluid quality?
To understand ourselves and our world, we need to understand how social systems function. Life evolved in complex natural environments, so I work with natural populations. I'm using proteomics to better understand how different kinds of ant societies control the development of the next generation. I analyze how these societies manage conflicts with the molecules they feed to their developing young.
We think that individuals in the colony may have different but complementary metabolic roles, and that the social fluid exchanges allow the components they produce to be transmitted across the colony's social network. This creates a metabolic division of labor. I'm trying to see if certain workers are specialized in building specific molecules that are used, for example, by the queen or by larvae during development. We hope to show that the ants in the colony cooperate like cells in a complex multicellular organism like us.
Ants are incredibly diverse, with 14,000 species showing a huge range of behaviors, morphologies, and lifestyles. What traits, like diet or colony size, lead a species to evolve trophallaxis? In species that engage in trophallaxis a lot, every individual is connected by the trophallactic network, and this fluid becomes an important means of communication and coordination. By looking at the proteins in the trophallactic fluid over many species, I am trying to understand how the social circulatory system has evolved and what it has evolved to communicate or transmit.
Sociality in bees, ants, and wasps evolved independently. What role does trophallaxis play in this evolution of sociality? In social wasps, workers feed larvae by trophallaxis, and larvae also give back trophallactic fluid to workers. Wasps offer a great opportunity to study the evolution of trophallaxis because these species exhibit the complete spectrum of social complexity, from solitary to highly eusocial.
So, social exchanges occur everywhere in biology, from bacteria to humans, and yet we understand so little about them. Part of that is because research in social fluids is compartmentalized in many different ways, in terms of the organisms people study, in terms of the type of exchanges. For example, people study milk or they study seminal fluid. But most problematically, the research is compartmentalized into types of exploration. There are people who come from medicine, people who come from agriculture, or people who come from evolutionary biology. So in my own work, I hope to really bring together people from these different fields and create this new field of social fluids. And I hope more broadly in science, we can do some of the same. Think of all that we could learn if we could get out of our silos, go outside, and do science in those in-between spaces.
So back to our creative expedition. The unknown is not partitioned into neat disciplines, and neither is curiosity. The way we do research and the way we support research needs to reflect that. We need to support open minds if the goal is to explore open spaces. Let's allow the breaking wave of our scientific understanding to seep into the open spaces between disciplines.